Hi, my name is Grace McCracken,
And I'm Kinsey Thompson.
and the article we were studying for this project was called Streptococcus pneumoniae G5 domains bind different ligands. And the authors for this article were N. Pakovich and J. S. Retzig, and it was published in Protein Science earlier this year. So just a little bit about the article. Streptococcus pneumoniae is a bacterial pathogen, and it uses G5 domains for a variety of survival factors. These survival factors include biofilm formation, as well as cell-to-cell adherence. And biofilm formation is really important because it has to do with disease reoccurrence. And so it's very important clinically to understand what allows these molecules to form these biofilms. So G5 domains are grouped as 5 conserved glycine residues in membrane-anchored surface proteins. And previously, they assumed that N-acetylglucosamine, or NAG, was the primary ligand used by these G5 domains. But this article sought to determine whether or not that was actually the true case. So this study also determined the different folding properties of G5 domains and how that impacted their binding modules and their adhesive functions. So S. pneumoniae has 5 G5 domains, 4 of which are single copy and were therefore studied in this article. And then a 5th domain came from Enterococcus faecium, which is a vancomycin-resistant bacteria, and is called VAN-W G5 throughout the rest of this study. So here's just a little graphic of the 4 S. pneumoniae G5 domains. You can see the double orange circles represent the conserved domains, and they're all single copy, so just one per S. pneumoniae. And then just a quick introduction to the materials and methods. Kinsey will mostly discuss these, but there was used protein expression and purification just to isolate the proteins using dialysis and centrifugation. Then there's also NMR and chemical shift assignments collected by a spectrophotometer, and this was used to determine structure, as well as the R1 and R2 relaxation rates and NOEs were also used to confirm the structure and find stereochemistry. And then finally, towards the end, NMR titrations and chemical shift perturbations came into play to determine the interaction between G5 and the ligands of NAG, as well as other potential ligands of heparin and zinc.
The first thing these authors did was compare the amino acid sequence of the selected G5 domains and assess their chemical shifts using the N15 heteronuclear single quantum coherence spectra. In the sequence comparison, as seen in the top of the figure, they found conserved acidic, which is red up there, basic, which is blue, and the hydrophobic, which are green. Those are all conserved residues between at least 3, if not all 5, of the G5 domains. They also showed the 5 glycine residues that were roughly conserved by the asterisks that Grace is pointing at. The chemical shifts showed a broad chemical shift in the S. pneumonia G5 domains, which are the first 4 that are displayed. That would be IgA1P, ENDD, BGAA, and SPR1403 G5 domains, and that's indicative of well-folded proteins, while the VAN-W G5 domain showed a narrow chemical shift, indicating it is an unfolded protein. There we go. They also noted that the SPR1403 G5 is especially prone to self-association. The IgA1P G5 and BGAA G5 moderately self-associate, and END G5 do not self-associate, which is important for the next steps of the experiment. Next, the authors looked more closely at the structures of IgA1P G5 and ENDD G5 to confirm that they have their typical G5 folds. They chose IgA1P because it is a commonly known virulence factor, and ENDD because it shows no self-association. They found the typical G5 structure in both of these proteins. The structure consists of 2 distinct beta sheets comprised of 3 beta strands each, with the 3rd strand woven between the 1st and the 2nd strands. and that is seen, the two sheets are the red, or the blue on top and the green on the bottom. The connecting regions between the two sheets were also found to be similar, consisting of 3 to 4 amino acid residues for each protein. There were a few differences observed, particularly of interest. The ENDD had a larger beta sheet near the C terminus, and the IgA1P had a larger beta sheet near the N terminus. They continued to look at the IgA1P and ENDD, but this time they looked at the dynamics of the G5 domains using R1 relaxation rates, R2 relaxation rates, and heteronuclear NOEs. The R2 relaxation rates were seen to be elevated in the loop regions, primarily at the ends of the G5 domains, as seen in the top of that figure for both sides. Then the R1 relaxation rates were seen to be slow, and the NOEs were the heteronuclear NOEs were small, and those two were particularly at the ends of the G5, the caps of the G5 domains as well. The R2-R1 ratio was mapped onto the NMR structures, which showed additional dynamic regions in the connecting region of the G5 domains, which is different than the other graphs had shown. The authors also noted that all the data for the IgA1P was collected at lower concentrations to ensure that the dynamics were due to intermolecular motion rather than self-association. Finally, they looked at the ligand binding capabilities of 3 of the G5 domains. They investigated ENDD and BGAA because they did not self-associate, and they also looked at IgA1P again because it only weakly self-associates. There were no CSPs observed in any of the G5 domains when NAG was present, which was a surprise because NAG was the proposed primary target of the G5 domains. They found that heparin induced CSPs which indicates it binds weakly to IgA1P, but not the other 2 proteins. They also looked at zinc, and that induced CSPs and bound all tested G5 domains at varying affinities. The binding affinities are as seen here. It bound strongly to ENDD-G5, and then next was BGAA, and the weakest was with IgA1P. They also looked at the binding isotherms of the ligands to the proteins in increasing concentrations, which is the bottom figures down here. Each color line indicates a different site-specific change that was made, which impacts the binding affinity. Finally, the authors noted that both heparin and zinc induce similar CSPs in IgA1P, leading these authors to believe that both ligands bind with the same beta sheet and may even have overlapping binding sites.
All right, so just to summarize some of the conclusions that we can draw from the results that we've heard so far, it was determined that the 4 S. pneumonia G5 domains are highly folded, while the Van W G5 domain was not, with the IgA1P being more self-associating. So 3 of the 4 S. pneumoniae domains did not engage NAG, which was proposed to be the primary ligand for these interactions. So that was a really surprising result that did not follow with the hypothesis of these experimenters. On the other side, all 3 of these engaged zinc. And so they have a proposed biological function for this. Since bacterial infections cause the host cell to create higher zinc concentrations during a part of the cytokine immune response. So it seems like the bacteria may have learned to exploit this higher concentration of zinc and learn to use that for self-association and adherence in the biofilm formation that we talked about at the beginning. These proteins also engage heparin in their G5 domains, especially IgA1P G5. And heparin is an anticoagulant, and they believe that in this case it basically just functions as extracellular DNA and sort of builds a structure for the S. pneumoniae to build its biofilm around, and so that's why it has that binding affinity. And if you take a look at this figure, you can see that the binding affinity to heparin is very high, based on the high CSP numbers here. And then S. pneumoniae G5 interactions likely play a role in biofilm formation and or epithelial cell adherence, which is what they had hypothesized at the beginning, and so that continues to be a trend through the entire paper. And then these G5 domains do not bind NAG, but rather interact with different ligands. So far we've discovered zinc and heparin, but more research is definitely required since this study found out that basically everything they understood about this was incorrect up to this point. So then one more look at this diagram. So you see the CSPs at the top here. As Kinsey talked about earlier, these are used to determine ligand interactions. So the IgA1P is both of these, and this one's IgA1P with heparin, and this is IgA1P with zinc. And so this bar graph shows that although the structure of IgA G5 domains indicates heparin and zinc bind in nearly the same location, they have to be driven by different interactions because the CSPs are very different. So if you see here, it's nearly the same location where heparin and zinc bind, and it seems to look similar, but the CSPs are much different, which implies that there's more going on than just the location of the binding. So that opens up another area for studies, what is actually going on in the binding there that creates the affinity. And then understanding that better will help scientists in understanding how to prevent reoccurrence of disease by learning how to disrupt the biofilm formation of the bacteria. And here is the reference for our paper.